Okay, so let's talk about blood. Actually, I don't know why this has been coming up, but a lot recently I have been discussing blood quite frequently. I've got a good friend of mine, their son has been having some issues, health issues, and so they have been doing a lot of blood draws on him. And so they're concerned about some of the things that we're going to talk about today, like his white blood cells, his platelets, and his red blood cells. So they've been concerned about some of those cells that are in the blood. So we are actually going to discuss some of those things. So there's at least some relevance for me. I hope there's some relevance for you and anyone else that's viewing this video. But let's talk about, we've talked about blood, believe it or not, in anatomy and physiology, biology 210, the predecessor to this course. And our first experience with our discussion on blood is that it is a connective tissue. And so there are three types of connective tissue, and blood falls under the third type, which is called fluid connective tissue, blood and lymph fluid. So this type of connective tissue is going to be a connective tissue that is constantly regenerating. Some connective tissues regenerate quicker than others. This one here is one of those. So think of our blood as like the supply line. This system here is going to carry not only the gases that we use, all right, for aerobic cellular respiration, like oxygen and then its waste product carbon dioxide, but it'll also carry fuel that cells need, like glucose, for example, or amino acids or fats, building blocks, things of that nature. So it's our supply line. And then also, as you know, just like us, we as humans, when we eat something, we're eventually going to dispose of it in the waste form. Well, our blood carries some of the cellular wastes around, and also it is an important transport system for hormones, those chemicals that you just talked about in chapter 17, those chemical messengers that are generated by certain tissues in the body that are going to affect other tissues in our body. So blood moves throughout our cardiovascular system. And so what moves it is the heart. That's the heart's job. It has one job, and that's just to pump the blood, to move it around throughout our body, through the pulmonary circulatory system, and then through our systemic cardiovascular circulatory system. So for the most part, when we're talking about, we'll talk more about this when we get into the cardiovascular system, that any tube-like structure that carries blood away from the heart, simple, it's an artery. Anything that brings blood towards the heart is a vein. And then where our arteries and our veins connect to one another in the tissues themselves, that's an exchange point, that's the capillaries. All right, the capillaries are the smallest of all blood vessels. So what's in our blood? Our blood is primarily made up of two things: formed elements, you'll hear me refer to them as the formed blood elements, and that's usually the cells, and then the plasma, that's the liquid that the cells are found in. So when we're dealing with this on a connective tissue scale, connective tissue is made up of three things. It's made up of cells, protein fibers, and ground substance. So in this situation, we know what the cells are. Those are going to be our formed elements there. And then our plasma is also going to be known as our GS, our ground substance. That's the media in which the cells usually reside. The cells usually produce the ground substance in this case. Right, but I mean, in most cases here. So our formed elements are going to be our red blood cells. They're responsible for carrying our respiratory gases, carbon dioxide and oxygen. Then we have our white blood cells known as the leukocytes. They're going to kill off any infectious agents, hopefully. And then our platelets, which help us to form blood clots, therefore preventing the loss of blood. And then plasma is the fluid part. And so that contains pretty much everything else. It's going to hold on to, all right, our formed blood elements, but everything else that we're going to discuss that we find in blood is going to be found within the plasma. So that will involve different proteins like albumin, antibodies. We'll also see some of our dissolved solutes. It could be anything from ions, right, to we'll have hormones dissolved in there, found in our blood plasma. So just think of the plasma as the liquidy portion. So what does our blood do? some of the functions here of blood. One, right, like I said before, it's our transportation system. It is going to transport things, right, throughout your body. And so you can pretty much, for the most part, you know this. If you've taken the first half of this course in 210, you kind of understand, right, when we talk about cells, how cells need nutrients to undergo some of their functions, right? So what gets those nutrients to those cells? A lot of times it's going to be the blood. So it's going to act as our transportation system. It's going to carry those blood cells, red blood cells, white blood cells, platelets, but also, all right, anything that might be dissolved in the blood plasma, certain molecules, certain enzymes, certain ions, sodium, potassium, calcium, all that fun stuff. And so essentially, when we talk about our respiratory gases, as you know, it takes oxygenated blood from the lungs into the heart, pumped out to the systemic system there, out to our tissues. And then from our tissues, we'll bring our deoxygenated blood, primarily the carbon dioxide will be found in that. And that's going to carry it back to the lungs to get exhaled off back into the atmosphere in exchange for more oxygen during inhalation. So a couple things when we're looking here for the transportation: nutrients, right, glucose, amino acids, fatty acids, hormones we talked about before. Heat is interesting. Your blood is about 1 degree Celsius warmer than your normal body temperature. Traditionally, on average, body temperature is 98.6 degrees, right? So the blood will usually be a whole degree warmer than that. So that plays a big role when we're dealing with homeostasis, when we're trying to establish, right, cold and hot environments, the direction or where the location of blood is going to be found in those tissues. If you're very cold, where does the blood go? It gets shunted away from your extremities more towards your internal organs because it helps to warm those organs and keep them warm, and vice versa. I mean, when you're exercising, you're outside in a hot environment like this time of year down here in South Carolina, okay, you'll start to sweat. That's a cooling mechanism. Also, though, we will transport blood to, all right, the cutaneous blood vessels in our skin, get it close to the surface of the skin, and we can give off heat that way also. So protection, that's primarily going to rest on our white blood cells here, right? Our neutrophils, our macrophages, right? The lymphocytes, all of those fun, and we'll talk about those blood cells. Those white blood cells are going to help us to fight off certain pathogens, right? The plasma proteins, like I said before, antibodies is a perfect example. We hear a lot about that, especially with COVID-19. A lot of folks were talking about antibodies. Well, a lot of our antibodies, right, can be found in our circulation. In fact, when you go and have an antibody test performed, they are going to draw blood and they're going to measure how much antibodies you have in your blood. And then we'll see also platelets, and then of course our plasma proteins that are going to be found in our blood, which will help promote clotting. Okay, so if you damage a blood vessel and you're losing blood, right, those plasma proteins will help with that clotting. All right, I already talked about body temperature. All right, body pH, this is very important. We'll get into that much later in this course. But of course, the normal pH range for our blood is 7.4. I hate when it does that. All right, so 7.4, which is slightly basic, a little bit more alkaline, right? But this is an ideal environment for most of our body's functions, chemical reactions to occur. A lot of our body physiology is optimal between 7.35 and 7.45, normally floating around 7.4. If we start to skew outside of that normal range, then we're messing up our homeostasis. When we're messing up homeostasis, then we're going to start to run into some issues, right? Especially if we become, for example, too acidic, all right? Then we get what's called denaturation of the proteins, and the proteins start to unfold. They start to unravel, so to speak. And when that occurs, when a protein is not functional, it's pretty much useless. And one of the functions of proteins is for chemical reactions, as enzymes. All right? So important that we maintain that pH. Our blood plays a big role. Fluid balance, we're going to talk again about this one later on in the course too, but this is important, especially when we're dealing with water. Right, so we'll talk about the normal amount of water to be taken in and how much you should be consuming on a daily basis. But our fluid balance is very, very important because we have these certain compartments within our body that we have to maintain a certain amount of fluid. And you've heard of the term dehydration, and that's a perfect example of what happens when some of our tissues do not have adequate water amounts. Well, how do we get fluids to those compartments? Through the blood. As you drink water, it's going to pass from the digestive system into your bloodstream, and then it can be circulated throughout. Now, if you have too much water, you've been drinking, say, 2 gallons of water a day, which is quite a bit, what usually happens? You have to go to the bathroom more often. And that's because your kidneys are working hard to take that water out of your blood, filter it into the kidneys in the form of urine, and then you can excrete it during urination there. So again, we'll talk a lot about that. But when we're talking about our fluid balance, that's what we're referring to, that exchange between our blood and then what we call the interstitial fluid. What's the interstitial fluid? Well, the interstitial fluid is the fluid that you find outside the cells. It's the fluid that's within the tissues. So you have the fluid that's in the cells. We call that the intracellular fluid, also known as ICF. And then you have what's known as the extracellular fluid. That's the fluid found outside the cells. But that includes the fluid that's in your blood, like your blood plasma there, what's in the blood plasma, and then also the fluid that's found within the tissues outside the cells. All right, so this is important. So we get to discuss that. So we're looking at our blood. Obviously, a lot of times when you've ever pricked your finger and it starts to bleed, it's red. So the color plays a big role in when we're talking about the physical characteristics of our blood. And so easy to remember, the color depends on the degree of oxygenation. What that means is just how saturated, how much oxygen is going to be in that blood sample. So when it's bright red, we know there's a lot of oxygen in that blood sample. When it is dark red, right, poorly oxygenated blood will have that dark red. If it's very, and it's interesting, carbon monoxide poisoning, although it doesn't happen terribly too frequently, I hope not, but folks that have carbon monoxide poisoning, if you look at their blood is described as a cherry red. And carbon monoxide loves hemoglobin, which is the protein that carries oxygen, way more than oxygen. So it'll bump the oxygen off. More about that later on. So normally, all right, when we're discussing the volume of blood in an adult, we're going to see about 5 liters, slightly less in females who are slightly usually just smaller body size than males, but about 5 liters in an adult. Right? So the next physical characteristic that we discuss is volume. And then we also have viscosity, how thick our blood is. All right, so blood is usually going to be thicker than water, right? I liken it to motor oil, right? So that's about 4 to 5 times thicker than water. And if you think about it, because of all the stuff that is in, all right, your blood, like I just talked about, those foreign blood elements, all right, red blood cells, white blood cells, platelets, that's going to add to the thickness of our blood. So really, our viscosity is dependent upon how much stuff is dissolved or suspended in our blood. So if you're going to go climb a mountain, right, and usually the elevation, right, the higher you go, the less oxygen is present. So how do we increase our transportation of oxygen in our circulatory system? We just make more red blood cells, more erythrocytes. But unfortunately, when we increase the number of erythrocytes, we increase the viscosity, make our blood thicker. That's what folks are going to experience when they blood dope. And then, of course, if you want to also increase the viscosity, become dehydrated, all right? Less water intake means there's going to be less fluid in the blood plasma. And by the ratio of the formed elements to the plasma, you'll have a higher number of formed blood elements to the fluid component of our blood. And so that is another way to increase the viscosity there, just to make your blood thicker. All right, and then our Our next grouping of physical characteristics of blood are going to be the plasma concentration of solutes, right? So when you go in and you get your blood drawn, and you look at your lab results, you'll see, all right, several things that are measured. Okay, we see how much red blood cells you have. We'll see how many white blood cells you have, what your platelet count is. But we're also going to look at, all right, the concentration of solutes in your blood, different types of proteins, different types of ions, sodium, potassium, magnesium, calcium, all that fun stuff. And so our plasma concentration of solutes is another important characteristic that we're looking at. And so that's going to help to drive the all-important process of osmosis. If you recall, osmosis is the movement of water from an area of high concentration to low concentration across what we refer to as a semi-permeable membrane. Well, the wall to the capillary is a perfect example. That's a semi-permeable membrane. It means it will let certain things pass. In this case, it will let water pass. But the direction of where water is going to move, is it going to move from inside the capillary to the tissues, or vice versa, from the tissues to the capillary? That plays a big role depending on how much fluid is available and the concentration of solids. So when we see in a state of dehydration, when our tissues are dehydrated, right, then our blood plasma becomes hypertonic because there's less water there. So there's a higher concentration of solutes. So that is going to draw water from the tissues into your blood. Because remember, water always wants to go from an area of high water concentration to an area of low water concentration. But when we're dealing with osmotic pressure, water is going to go to where there's a higher solute concentration. So if there's more solutes in the blood, then water moves from the tissues into the blood. And when it draws it out of the tissues, you become dehydrated. All right, we already talked about temperature, 1 degree warmer. And then, of course, we already talked about blood pH. All right, slightly alkaline, we're looking at 7.4. All right, and we definitely want to avoid denaturation, where we just ruin our proteins. All right, so let's look at the different components of blood. And when we do that, we are going to utilize a centrifuge, which is this machine. I think I have a picture of it. There it is. There's our centrifuge. So we draw the blood from somebody, right? We put it in a vial, depending on what we want to do with that vial. There's different colored tops, depending on what we've added to the vial. Sometimes we don't want the blood to separate. In most cases, we do want it to separate. So anyways, we take that vial, we put it in a centrifuge, we spin it for 10 minutes, and it separates into something that looks like this to our right. So we'll see on top this light kind of coating on top, and then we'll see this big red coating on the bottom. And then in between these two layers, we'll have this small, tiny, what we call a buffer coat. And so I'm going to talk to you folks about what is in, all right, these components here. All right, so when we're looking here at our centrifuge blood, that bottom coat, that red layer, that is the red blood cells. And so a little bit less than half, about 44%, are going to be primarily made up of erythrocytes, the red blood cells. And just in case you want to know, the average lifespan of a red blood cell is 120 days. You should know that. $120 days. So there's another example as to how our fluid connective tissue of blood is constantly regenerating. And it's because of that characteristic of, well, one of the characteristics of red blood cells. Then that middle coat that I was telling you about, that's the buffy coat. That's where we're going to see our white blood cells. And that's where we're also going to see our platelets. And then on top, that, I won't call it a clear coat, right? But that reddish, what we call straw-colored liquid, that is going to be the plasma, the liquid portion of our blood. And that is just over about half, about 55%. So we're going to talk about now what is in. If you look here, you can really appreciate the erythrocyte layer is pretty easy. It's just composed of the red blood cells. And then the buffy coat, again, that's going to be all your white blood cells and the platelets. But if you look here at the plasma layer there, look at all those different things that are in the plasma layer. Predominantly, the number that should be jumping out at you is this number. 92% of your plasma is going to be water. Right. Then our remaining percentages, roughly about 7%, are going to be proteins. And here are all the different types of proteins: albumins, globulins, and globulins. In that category, we include our antibodies. Right? Fibrinogen, that is a protein that helps with clotting. And then we have a series of regulatory proteins, which is slightly less than 1%. But then when we're looking at our 1% of other solutes, that's where we start to get into those ions and those different solids I was talking about, electrolytes. That includes sodium, potassium, right? Different nutrients, not including our respiratory gases, and then our waste products like ammonia, all right, or urea. So again, you can see that our blood carries quite a bit of stuff. So when we undergo this process, the centrifuge process, there's a term here, hematocrit, and that refers to the overall percentage of all the formed elements. Now, primarily, when we're looking at hematocrit, if you've ever looked at your blood results when you have a lab draw, a blood draw done at the lab, one of the values they put up there is hematocrit. So most doctors, when they're looking at this, they're only pretty much taking into consideration the erythrocytes, right? But it does include all the formed elements. But when we're looking here, I mean, look, less than 1% of your formed blood is going to be the white blood cell. So you can see how primarily we're going to just be judging the erythrocytes, the red blood cells, all right, when we're dealing with the clinical portion of hematocrit, primarily focusing on erythrocytes. So again, with adult males, and again, that is due to our androgen, our testosterone production, which influences, and you should remember what erythropoietin is. Erythropoietin is that hormone that's secreted by the kidney that is going to increase the production of red blood cells, hemopoiesis. It's going to stimulate your red bone marrow to make more red blood cells when you have a high oxygen demand. So you can see hematocrit levels should be higher in males, anywhere from 42 to 56, and in females, they'll have a smaller number, anywhere from 38 to 46. All right, so let's take a look at our blood plasma. Right, like I mentioned before, you can see here just by looking at the percentages, water is the major percent of blood plasma, followed by plasma proteins, then followed by our dissolved solutes. And so we'll talk more about this later on in this course, but plasma is an extracellular fluid. If you look at that term, extracellular, that is referring to the fluid that's outside of cells. But there are two compartments when we're dealing with extracellular fluid. One is interstitial fluid. Think of that as the fluid that's found within the tissues there. The other compartment is what we're talking about today, the blood, our blood plasma. All right, so that is why we are going to see in our interstitial fluid, when we compare it to our blood plasma, they're going to look very similar, except our blood plasma is going to have a higher protein concentration. And the reason why is because proteins, all right, in a healthy individual should stay in the blood. Not all proteins, okay, but the proteins that are circulating in your plasma primarily should stay in your blood in a healthy individual. Okay, so they're gonna, and one of the reasons for that is you want to have a higher protein concentration in your blood when we deal with oncotic osmotic pressure. This will help to draw fluid from the tissues into your blood plasma. All right, which leads me to this next part here. And this is what I want to discuss when we're talking about blood in general. So there's a term here at the top of the screen here that you see. Blood is a colloid. Right? And colloid is basically proteins. Right? So basically what that statement means is our blood, primarily the plasma, is going to contain proteins, colloids. And so because of that, it's going to generate an osmotic force, right? A pull, a draw on water. Right? And so we want this to occur, right, in normal, healthy individuals. We want to keep our plasma proteins in our blood. Right? So albumin, globulins, fibrinogen, tons of different type of clotting proteins, several types of enzymes that you'll see for certain physiological chemical reactions, and some of the hormones that are going to be circulating through, all right, our bloodstream. Those are going to make up our plasma proteins. Now, primarily, most of those plasma proteins are created, all right, by this wonderful organ, the liver. But others, not all, all right, will be, obviously, you know that some of the hormones aren't going to be produced by the liver. They'll be produced by different glands, the adrenal glands, the thyroid gland, right? So again, liver doesn't produce everything, but it produces most of those. Right? So this is very important, right? So these organs are going to make these proteins and dump them into our blood. And so these proteins, when they're dumped into our blood, they're going to create what's known as the colloid osmotic pressure. This has to do with what we call bulk flow, right? But for right now, I want you to consider these plasma proteins. They're going to create and exert this colloid osmotic pressure. And what it does is it helps to hold fluid, or it prevents the loss of fluid from the blood into your tissues. So it helps, basically, what I could be saying is colloid osmotic pressure helps to prevent blood vessels, primarily capillaries, from being leaky, from letting stuff leak out of the blood vessels. That shouldn't be leaking out of the blood. I mean, we want oxygen to leak out into the tissues, and we want, all right, carbon dioxide to leak into our capillaries, because we got to get it back into the blood. We got to circulate it back up to the lungs so you can exhale it off back into the atmosphere. You know, those trees and those flowers and plants need that carbon dioxide. It's important. So this colloid osmotic pressure is terribly important in maintaining what we call the blood volume. You're going to find out that blood volume and blood pressure are heavily reliant on each other, mainly blood pressure on blood volume. If you increase your blood volume, you're going to increase your blood pressure. And if you decrease your blood volume, most likely you'll decrease your blood pressure. And you talked a little bit about that in chapter 17, right, when we were talking about aldosterone and ADH. And we'll talk more about that when we get into kidneys in much more detail. So don't worry if you don't have that quite mastered, all right, but we'll get you there. We'll get you there. All right, so certain diseases can mess with your colloid osmotic pressure. And as a result of certain diseases, for example, kidney disease, let's say, you have certain kidney diseases that affect the kidneys, damages the kidneys, and then the kidneys can't filter out the blood appropriately, and some of those plasma proteins will leak out of the kidneys into your urine, and you'll get rid of them. So as a result, all right, of some of these proteins leaking out of these tissues here, of the capillaries, what we'll see is they'll leave the capillaries and move into the tissues. And when they do that, water follows. I want you to think of water as the big follower. Okay, it gives into peer pressure. Water loves to follow salt, sodium. It loves to follow sugar, glucose. Right? Well, it will also follow certain proteins. So proteins are leaking out of the capillaries into the surrounding tissues, water follows, and the tissues start to swell. And so what do we call that? Edema. And you'll see it in people's legs most generally. You'll see an increase of swelling in those tissues in the legs. And so you should start to think all right, there's some issues here going on, right, with the circulatory system, but I can't rule out, right, I cannot rule out other tissues, organs like the liver, that sort of thing. So keep in mind. All right, so albumins are, all right, the most abundant group of plasma proteins. And you can see how large of a number, it's 58% here. And so they're primarily going to be responsible for that colloid osmotic pressure. So if colloid osmotic pressure overall starts to drop, you start to see symptoms like I was telling you, that swelling in the tissues, you should be thinking albumins first. Something's going wrong. The albumin production could be low. That's why we'll draw the blood just to confirm our diagnosis, right? So if we get our lab results back and we see that the albumins are low, then we should start to think, okay, there's most likely some sort of liver condition going on here. And so we'll check some of the other enzymes to confirm that diagnosis. Then we can run some imaging tests to see what's going on. All right. So albumins play a huge role, all right, in regulating that colloid osmotic pressure. They also play a large role in transport. So they're going to be responsible for transporting the different types of lipids that are involved, that you'll find in the blood. Hormones too. We talked about in chapter 17 how there's fat-soluble, water-soluble hormones, different types of hormones. Some hormones can dissolve right into the blood plasma and go throughout and be transported. Others need a transport protein. Albumin takes over for that. Ions too, okay? Calcium, sodium, some of those, right? Calcium has helpers, right? Certain proteins which will help, calmodulin, calsequestrin, those are a couple, and you remember those from chapter 10, all right, when we were talking about muscles. And so some of these hormones, some of these proteins act as transport proteins for certain ions. All right, globulins. Okay, this is the second largest. All right, we have different classes of globulins, our alpha globulins, our beta globulins, gamma globulins. All right, but the alpha and the beta, again, are going to act as transport proteins here. So they're going to transport the water insoluble molecules. Because keep in mind, all right, what does that even mean? Why do I even care if something's water insoluble or water soluble? Right? Well, if certain molecules are water insoluble, like oil is water insoluble, right? So we have these transport proteins that will attach onto those molecules so they can move in our blood plasma without any issues whatsoever. Then we have our gamma globulins, the real famous ones, the immunoglobulins, also known as antibodies. Okay, plays a huge role in defense. IgAs, IgGs, IgMs, right, IgEs, all these, the IG stands for immunoglobulin, and then the letter stands for the class there. So the gamma globulins fall under that. Fibrinogen, right, main function, blood clot. Right, so eventually fibrinogen is going to go through a series of different enzymatic or chemical reactions, and it's going to wind up as fibrin. And it's that fibrin which basically creates that net that will eventually, all right, clog up or stop blood from leaking out. It will essentially clot that area that was damaged, all right, and the fibrinogen plays a huge role in that. So when you talk to folks that are discussing, all right, when they've had their blood drawn and they're selling plasma and things of that nature, you may have heard the term serum, right? And we're not talking about the serum, the truth serum, or anything like that. When we're talking about serum, basically serum is going to be your blood plasma with the clotting proteins removed there. And then we have our regulatory proteins. Again, these are some more enzymes and some other hormones involved. All right, so we talked about blood and its components and the different things that you're going to find inside of them. But this goes back to And I got to tell you, I was very, very proud. I have an 11-year-old daughter. She's in 6th grade, and I think she was either in 4th or 5th grade. Maybe I think it was when she was in 5th grade. And she was telling me what she was learning in science. And she actually explained to me what a solute and what a solution is. And so I'm happy to say that she explained it very well. And if she were here right now, not a karate class, I would have her explain, all right, blood as a solution. But blood is a solution. And basically, you knew this subconsciously because I told you that there's lots of things in our blood that are dissolved in the blood. And so the solution is going to be that stuff that has other things that are dissolved into it. So that's what we're going to be discussing here. All right. So blood, being a solution, is going to have both organic and inorganic molecules, including ions, dissolved right into the blood plasma. And so when we talk about some of the respiratory gases, you'll learn when we get into that chapter that you'll have some oxygen and carbon dioxide attached to the hemoglobin molecule, but some will also still be in the solution there. All right, electrolytes. Again, this goes back to what I was telling you. When you get your blood drawn, you're looking at your calcium levels and your sodium levels and whatnot. That is going to be, all right, what we consider the electrolytes, and all of that will be dissolved into the solution. Now, polar, or if it's charged substances, they will dissolve easily. The non-polar are going to require our carrier proteins. So polar substances are going to dissolve easily. And the reason why is, this goes back to chapter 2 when we were talking about that biochemistry, all right, hydrophilic versus hydrophobic. Right. So the polar are hydrophilic. They're water loving, and they'll dissolve right into that blood solution because remember, plasma, 92% of it is water. Non-polar is hydrophobic. And so that's why it needs those carrier proteins. So the all famous process of making the components of our blood when we're discussing the formed blood elements is called hemopoiesis. Mentioned this before, all right, in 210 when we were discussing about bone in chapter 7. Right, but now we're going to discuss it in more detail, the actual process of hemopoiesis, right? And then that we'll finish up with the hemopoiesis today. I don't want to keep you too long. All right, so hemopoiesis, how do we make, all right, the formed blood elements? How do we make red blood cells? How do we make white blood cells? How do we make the platelets here? Well, this occurs in our primary lymphatic tissue in our bones, and it has to be red bone marrow. So we're going to start off, right, with hemopoiesis. We're going to start off with stem cells. So we give them a special name, hemocytoblasts. So you're going to hear this term right here called pluripotent. And pluripotent means that these cells can turn into almost any type, well, I said almost, right, any type of cell. Okay, so these cells are going to differentiate into a specific type of cell line. It could be a red blood cell line. It could be a white blood cell line, right? But it has an option. In this case, we have two, all right, lines. We have what's called the myeloid line and the lymphoid line. All right? Our myeloid line is going to be our red blood cells, all of our white blood cells except for the lymphocytes and the megakaryocytes. All right. Those are going, and our megakaryocytes, you should be thinking platelets. And then our lymphoid line is going to be only lymphocytes. So how am I ever going to remember that? Easy. What is the lymphoid line? Only lymphocytes. Myeloid line is everything else. That's how I remembered it. Okay, so lymphoid line, only the lymphocytes. Myeloid line, everything else. So how do these cells know when to turn from one cell type to the next? Well, no problem. We've got these chemicals, and we call them colony stimulating factors. And so these cell chemicals will act on these cell lines, and they'll basically direct them in the direction that they want to go. Now, it's not important that you know the specific colony stimulating factors. I'm not going to ask you to do that because I'm going to show you a picture here in a second. Where are you, picture? Here it is. And so you can see here on this diagram here, right, we have our pluripotent cell right here, the hemocytoblast. And like I told you, it can go into one of two cell lines. If it's going the lymphoid line, easy, it's only going to become a lymphocyte. If it goes the myeloid line, then it's got several different, all right, choices. But you can see here, these little boxes here are telling you, all right, the colony stimulating factor. And they vary. Like I said, I will not require you to know, all right, which colony stimulating factor is going to produce what. Right? You don't have to worry about that. All right, so let's start off here with erythropoiesis. And the name tells you what we're going to be making, erythropoiesis. So if you're going to bake a cake, you're going to have certain ingredients. Same thing here with erythropoiesis. We need certain ingredients, all right, besides the normal building blocks, obviously, to construct almost anything organic in your body. So when we're dealing with hemopoiesis and we're talking about the actual production of erythrocytes, we need iron. Guess why? Iron, which is Fe. Its chemical symbol is Fe, and that stands for ferrous. We need that for our hemoglobin. B vitamins, particularly B12, and amino acids. Because what do amino acids make? Proteins. And what do proteins make? A lot of stuff. Okay, so that's what we need. Iron, B vitamins, all right, specifically B12, and then amino acids. So again, I don't expect you to memorize all these different, all right, stages of erythropoiesis, but there's a couple things that I definitely want you to know. I'm going to point those out. So we're going to start off with our stem cell, the myeloid stem cell. And so that multi-CSF will be produced. It's going to stimulate the myeloid stem cell, okay, to move into our progenitor stem cell line. Right. So as you go through, I'm not going to walk through all of these, but I definitely just want to show you, you can see, right, we're going to start to march our way through these various stages of red blood cell, all right, production. So you'll notice we have a nucleus here and a nucleus here. When we get all the way to the bottom, our red blood cell does not have a nucleus. It is anucleate. So that means there's no nucleus in the adult form of the erythrocyte. It's one of the few cells that does not have a nucleus. And that is partly the reason why red blood cells, right, only live for 120 days on average. So we have different names, all right, for this cell as it moves through. But one of the ones I want to show you is these last two. Okay, a reticulocyte. A reticulocyte is what we refer to as a baby red blood cell. So when you've gotten your blood drawn, if you see that there's an increase in reticulocytes, one of the things, there's several possibilities, but what a lot of clinicians think when they see that is, the red bone marrow is making a lot of new red blood cells. And a reticulocyte is just a baby cell, okay? It doesn't have the organelles, right? It does have ribosomes. What do ribosomes do? They make protein. What's the important protein in the red blood cell? Hemoglobin. Because that's what oxygen attaches itself onto. But when we get to the adult form, the erythrocytes, we no longer have ribosomes. So we don't have a nucleus and we don't have ribosomes. So that cell is not making anything. It's not making anything. All right, so that's what we're looking at here. Just a quick picture, all right, when we're dealing with erythropoiesis. Okay, so let's talk about our next type of hemopoiesis, and that's leukopoiesis, the production of leukocytes. So there's three types of leukocytes, right? We have granulocytes, monocytes, and our lymphocytes. Well, we already talked about the lymphocytes here. We're not going to deal with lymphocytes. That's somewhere else here in a moment. So what I want to talk about are the granulocytes and the monocytes. Right? Our granulocytes, if you look at the first four letters, gran, right? Think of granules, dots like sand. Right? So the three types of granulocytes are neutrophils, basophils, and eosinophils. And it's not important about the colony stimulating factor, but understand that we are going to release multi and GM CSF, and that is going to steer us, our myeloid cell, right, into our progenitor cell, which is going to then eventually, right, when we're dealing with granulocytes, that's going to steer us toward what we call the myeloblast. Right, now, when we're dealing with the monocytes, right, those come from our myeloid stem cell line. Right, same thing what we're looking at. We're going to have our colony stimulating factors here, which are going to steer our cell right into this cell lineage. And we're going to start off with the monoblast, which will eventually, a monoblast will give us a monocyte. A monocyte is the circulating blood, the blood circulating form of a macrophage. So I'll show you. Zoom in here. All right, so here's our leukopoiesis here. Right, again, if we're going to make a lymphocyte, we're going to move towards the lymphoid line, but when we're dealing with our granulocytes and monocytes, we're going to be steering towards the myeloid line. So we go through the myeloid stem cell, then we have our multi-CSF, which is going to steer us here, all right, and generate our progenitor cell with the help of our GM-CSF, and then depending on which type of colony stimulating factor, easy to remember, G is for granulocyte, M is for monocyte. So we have the G-CSF, we're going to form our myeloblast. Again, I won't bore you through all that, go through a series of enzymatic reactions, and it's going to yield us either eosinophils, basophils, or neutrophils. Again, there's little granules in each of these. That's why they fall under the granulocyte spectrum here. Then, if we're dealing with our monocyte line, okay, eventually we're going to produce our monocyte here for blood circulation. But when a monocyte leaves the blood circulation and moves into the tissue, that's when it turns into a macrophage. You should be familiar with the macrophage. That's the Pac-Man white blood cell. Okay. All right, so that's leukopoiesis. All right. Our leukopoiesis, when we're dealing with our lymphocytes, which come from that other stem cell line, the lymphoid stem cells. Again, same thing will happen, right? We are going to, and our end game with our lymphocytes is going to be our B lymphoblasts and our T lymphoblasts, which are also known as B cells and T cells. And so these cells, they'll mature into the B lymphocyte and the T lymphocyte. And then we'll also see, all right, from the lymphoid stem cell line, we'll also get a really cool cell, which we'll learn more about in the immune system chapter, are natural killer cells. And that's exactly what they do. Their job is to go out and hunt things down and kill them. So here you can see, right, when we're looking at our lymphoid stem cell line, right, you got your lymphoid line here, which is either going to be B cells and T cells, and they'll eventually yield us the B lymphocyte and the T lymphocyte, or we can go directly from the lymphoid stem cell directly to our natural killer cell. And then we can't forget about our platelets. The platelets are the last ones here, and that the term that we give to platelet production is thrombopoiesis. So we're going to start off with the megakaryoblast. So this is going to be our myeloid stem cell. And the megakaryoblast is eventually going to yield us what we call a megakaryocyte. And a megakaryocyte is this huge cell, right? Huge, huge cell. And what it does is it kind of migrates towards the edge of the tissue there, okay, your blood vessel, and it has these little extensions that kind of extend off the main cell part itself, right, which we call these proplatelets. I'll show you an awesome picture. So they'll extend through the blood vessel wall into the bloodstream. And then as the blood is whooshing by these proplatelets, it just tears off slices, these fragments. And those fragments become the platelets. I'll show you a good picture here in a second, but let me just show you. Here's our thrombopoiesis here, okay? Thrombopoiesis, it goes through a series, all right, of all these changes. We eventually get down to our megakaryocyte. And the megakaryocyte has these long extensions, and that's called the proplatelet. And it sticks that long extension out into the bloodstream, and as the blood is whooshing by, it just tears off pieces of it. And those pieces that are torn off, those are your platelets there. I'll show you a nice picture here. All right, so here are your megakaryocytes. And so those megakaryocytes, right, will stick those extensions into the bloodstream. And as the blood is whooshing by, it just tears it off. So that's why when you're looking at what they call a peripheral blood smear, when they take a drop of your blood and put it under a microscope and they look at all the different cells there, the smallest little dots on that peripheral blood smear, those will be the platelets. They're tiny. All righty. I think I'm going to stop here. And